Good morning, everyone, and welcome. I'm delighted to have you here today. Today's presentation is centered on prospective validation of sterile manufacturing processes. I'll be walking you through the key concepts, the importance of validation, and the best practices in sterile manufacturing. We'll explore how these processes ensure product safety and compliance. So, let's dive in and explore this vital topic together. Prospective validation is a crucial part of the pre-market process. It involves evaluating, controlling, and proving that manufacturing processes, especially sterile operations, can consistently deliver high-quality products before we start routine production. This step helps ensure that everything is working as it should and that the process is reliable. It's also a key requirement under good manufacturing practice standards, particularly for sterile products. This is because maintaining aseptic integrity and sterility assurance is vital for patient safety. When we talk about prospective validation, we're essentially building confidence that the manufacturing process will produce safe and effective products every time, which is fundamental to our overall goal of protecting patient health. In this slide, I want to highlight the main objectives of prospective validation. First, it's essential to establish documented evidence that our sterilization or aseptic processes reliably produce sterile products. This means we need solid proof that our methods consistently meet the required standards. Next, we must confirm that critical process parameters, or CPPs, and critical quality attributes, known as CQAs, stay within the validated limits throughout the process. Ensuring this consistency is vital for product quality. Additionally, we need to verify that our sterilization and aseptic manufacturing systems are reliable, repeatable, and robust. This gives us confidence that the process will perform as expected under various conditions. Finally, it's important to demonstrate compliance with regulatory guidelines, including EU GMP Annex 1, WHO TRS, US FDA standards, and PICS. Meeting these requirements not only ensures regulatory approval, but also reinforces our commitment to quality and patient safety. Overall, these objectives are about building a strong, validated foundation for our sterilization processes that uphold quality and safety standards across the board. Let's start by understanding the product characteristics that are critical for sterile operations. These include factors like pH, viscosity, sensitivity to heat, and the risk of contamination. Each of these elements can significantly impact how the product behaves during processing and how we need to control the environment to ensure sterility. Next, we need to consider the compatibility of the container closure system. It's essential that the container and closure work seamlessly together to maintain sterility and prevent leaks or contamination throughout the product's shelf life. We also have to identify the sterility-critical steps in our process. These are the points where any deviation could compromise the sterility of the product, so understanding and controlling these steps is key to successful validation. Finally, I want to highlight the key elements of prospective validation in sterile operations. This involves planning and executing validation activities before production begins to demonstrate that the process consistently produces a sterile product. It's about ensuring quality and safety from the outset, which ties back to our overall goal of maintaining high standards in sterile manufacturing. Now, let's dive into how we use FMEA and HACCP approaches to identify potential risks in our processes. First, we look at microbial contamination risks, which are critical because they can directly impact product safety. Then, we examine environmental contamination risks, such as cross-contact or environmental sources that could introduce hazards. We also consider human interventions and critical manipulations, because human actions are often a point where things can go wrong if not properly controlled. Next, we evaluate equipment failure or process deviations, since equipment issues can lead to contamination or inconsistent product quality. The goal here is to pinpoint where things might break down so we can implement effective control measures. From there, we determine critical control points, or CCPs, which are specific points in the process where controls are essential to prevent or eliminate hazards. And associated with these CCPs are our control strategies, practices and procedures we put in place to keep risks at bay. All of this fits into our broader risk assessment, a systematic way to evaluate and prioritize hazards based on their severity and likelihood. By doing this, we ensure that our food safety measures are targeted and effective, ultimately supporting the overall integrity of our food safety management system. When developing a detailed protocol, it's crucial to clearly define several key elements to ensure robustness and consistency. First, you'll want to specify the scope of the validation strategy, outlining exactly what processes and parameters are being validated. This includes establishing your batch size and determining the number of batches needed, which typically involves at least 3 consecutive successful batches to confirm reproducibility. Next, you'll need to develop a sampling plan along with acceptance criteria. These define how and when samples are taken, and what results are deemed acceptable. In-process control parameters are also essential. They help monitor critical points during manufacturing to catch any deviations early. Additionally, sterilization cycle parameters must be precisely defined to ensure consistent and effective sterilization across batches. Testing requirements form the backbone of validation, covering sterility, endotoxin levels, particulates, fill volume accuracy, bioburden, and other relevant quality attributes. All these elements should be incorporated into your validation protocol, which serves as a comprehensive plan for how validation will be conducted. Remember, preparing a thorough validation protocol is fundamental to the overall quality assurance process. It provides a clear roadmap, ensuring that every step is documented and reproducible, ultimately supporting product safety and efficacy. Now, let's discuss sterilization validation, starting with filtration sterilization, also known as sterile filtration. This involves conducting filter integrity studies both before and after the filtration process to ensure the filter is functioning correctly. We also perform microbial retention validation to confirm that the filter effectively removes microorganisms, which is critical for product safety. Additionally, worst-case challenge studies are carried out to test the filter's performance under the most challenging conditions, ensuring robustness. We also evaluate filter compatibility and extractable studies to verify that the filter materials do not leach harmful substances into the product. Moving on to terminal sterilization validation. Whether using autoclaves or dry heat sterilizers, this process involves several key studies. Heat distribution and penetration studies are essential to confirm that sterilizing heat reaches all parts of the product uniformly. Biological indicator challenge studies are used to verify the sterilization process's effectiveness. These indicators contain resistant microorganisms that must be reliably inactivated. Hold time verification ensures the product is maintained at the sterilization temperature for the appropriate duration. Load pattern qualification confirms that the way items are arranged allows for effective sterilization. Lastly, F0 value or lethality determination quantifies the sterilization process's effectiveness, providing a measurable parameter to ensure consistent sterilization. All of these steps are part of prospective validation in sterile manufacturing, which is crucial for maintaining high-quality standards and ensuring patient safety throughout the production process. Now, let's talk about the simulation of the entire aseptic filling operation using sterile media. This process is designed to mimic real-world conditions as closely as possible. We focus on worst-case scenarios, which means testing under maximum interventions and the longest durations to ensure robustness. The key acceptance criterion here is zero contamination for critical units, as any contamination could compromise product safety. These media fills are typically conducted twice a year, following prospective validation, to verify that our processes remain under control. This aseptic process simulation is a vital part of our quality assurance. It confirms that our sterile environment and procedures can consistently prevent contamination, even under challenging conditions. Overall, these simulations help us maintain high standards and confidence in our aseptic operations. Now, let's discuss some critical aspects of our cleanroom validation process. First, HVAC validation is essential. This involves verifying airflow patterns, pressure differentials, and ensuring the integrity of HEPA filters. These elements are the backbone of maintaining a controlled environment, preventing contamination, and ensuring product safety. Next, we have PQ, or performance qualification, specifically for cleanrooms. This includes both viable and non-viable monitoring. Viable monitoring checks for microbial presence, while non-viable monitoring tracks airborne particles. Together, these tests confirm that the environment consistently meets cleanliness standards. Personnel qualification is another vital component. Proper gowning procedures and aseptic handling techniques are fundamental to prevent introducing contaminants. Ensuring staff are well-trained and qualified directly impacts the overall integrity of the cleanroom. We also perform microbiological verification on surfaces, air, and equipment. Regular testing helps us detect any microbial presence early and address issues before they affect product quality. All these aspects, HVAC, PQ, personnel qualification, and microbiological testing, are interconnected. They form a comprehensive validation strategy that supports our overall goal, maintaining a sterile, controlled environment to ensure the safety and efficacy of our products. Now, let's talk about the importance of IQ, OQ, and PQ for critical equipment like isolators, RABS, filling machines, sterilizers, and washers. These validation stages, installation qualification, operational qualification, and performance qualification, are essential to ensure that each piece of equipment functions correctly and consistently within our process. They help us verify that everything is installed properly, operating as intended, and performing reliably over time. Moving on to utilities validation, this is just as crucial. Purified water and WFI are used extensively in our processes, so confirming their quality and supply consistency is vital. Clean steam, for sterilization and other applications, must meet strict standards. Compressed gases, including nitrogen and clean air, are often used in filling and sterilization, so their purity and supply reliability need validation too. Overall, equipment and utilities qualification are foundational elements of our quality system. They ensure that every part of our operation, from equipment to utilities, is working correctly, safely, and in compliance with regulatory standards. This, in turn, helps us maintain product quality and patient safety throughout the manufacturing process. Now, let's talk about the execution of the approved protocol. It's crucial that trained personnel carry out all procedures to ensure consistency and compliance. We need to document every process parameter and test result meticulously, as this creates a transparent record of the process. Managing deviations is also key. When something doesn't go as planned, we conduct a root cause analysis to understand and address the underlying issues. Additionally, we review microbiology, sterility, and product quality data regularly to verify that everything meets the required standards. All these steps are essential parts of validation execution, ensuring that the process is reliable and reproducible. Together, these elements help us maintain high quality and regulatory compliance throughout the manufacturing process. Today, I want to highlight some of the typical acceptance standards we use to ensure quality and compliance. First, successful completion of 3 validation batches is essential. This helps us confirm that the process is reliable and reproducible. Next, all sterility-critical parameters must stay within predetermined limits. This is crucial to maintain product safety. We also look for no contamination in media fills, which are simulated runs to detect any potential issues. Additionally, 0 failures in sterility and endotoxin tests are required, ensuring our products meet the highest safety standards. Lastly, consistent results in environmental and personnel monitoring confirm that our cleanroom conditions and staff practices support our quality goals. These standards are the backbone of our quality assurance process and help us deliver safe, effective products every time. Now, let's discuss what the final report should encompass. First, we need a clear summary of the results compared to the acceptance criteria, so we can easily see if everything meets the set standards. Next, a statistical evaluation of the data trends is essential. This helps us understand patterns and variability within the data. We should also include any deviations observed during the process, along with the corrective actions taken to address them. It's important to draw a conclusion on the overall process suitability and robustness, ensuring that the process is reliable and consistent. Additionally, we should make recommendations for routine control measures and revalidation schedules to maintain quality over time. All these points come together in the validation report, which documents the entire validation process comprehensively. By covering these areas thoroughly, we ensure the report provides a complete picture of the validation effort and supports ongoing quality assurance. Prospective validation is really just the beginning of the life cycle. Once you've established that initial validation, the real work starts. It's about continuous monitoring, keeping an eye on environmental conditions, equipment, and process parameters to ensure everything remains in control. We also need ongoing process verification, like OPV and CPV, to make sure that the process continues to perform as intended over time. This isn't a one-and-done deal. It requires periodic review and revalidation to adapt to any changes that might occur. Change control and deviation trending are also critical. They help us identify, evaluate, and address any deviations or changes, ensuring that the process stays in a validated state. All of these activities tie into life cycle considerations, thinking of validation as a continuous, dynamic process rather than a one-time event. This approach ensures quality, compliance, and consistency throughout the product's life cycle, aligning with our overall goal of maintaining high standards and continuous improvement. Prospective validation plays a crucial role in ensuring the highest level of sterility assurance in our manufacturing processes. It helps us minimize contamination risks by thoroughly testing and confirming that our methods are effective before production begins. This approach establishes a scientifically sound foundation, which is essential for meeting regulatory requirements and ensuring reproducibility. When we validate prospectively, we're basically setting the stage for a consistent, reliable process that produces safe and effective sterile pharmaceutical products. In summary, this step is vital for building confidence in our manufacturing quality and ultimately delivering safe medicines to patients. For further support, please contact us. We're here to help with any questions or additional information you might need. Thank you so much for your time and attention today. Remember, we're committed to assisting you in any way we can, so don't hesitate to reach out. We appreciate your engagement and look forward to working together.